Hey team, Dr. Jake Auty here, and in this video we're going to jump into the cells of the innate immune system. So the basic strategy is the previous video I gave you a rough overview of the immune system. Now we're going to talk about some of the parts of the immune system, like the cells of the innate immune system, and in the next video the cells of the adaptive immune system. But then in future videos we're going to try tie it all together, because we need to step back and look at the big picture of the immune response. So in the future videos we're going to say, let's say you get a bacterial infection, how do all these cells and parts of the immune system interact to create an immune response to the bacteria? So if you feel a bit confused and a bit worried that you're taking on too much information all of a sudden, don't worry, because we're going to go over it a couple more times to really see how these players all interact to generate our innate immune response. Now, this little background image here is a great, probably the most important immune cell. That's just because it's the one I study. It's a macrophage, a big eater, and in here it has these fluorescently labeled bacteria. So this red macrophage has eaten some green bacteria, creating these yellow dots inside its cytosol. Anyway, beautiful image. Let's jump into it. So where are these immune cells? Well, we have circulating immune cells that live inside of our blood. So here's just two here flowing through our blood vessel. So these are called circulating immune cells. We then have a lymphatic system. Now, you don't hear a lot about the lymphatic system. You know all about the arteries taking the blood away from the heart and the veins taking the blood back to the heart. And that is the circulatory system, and it's all driven by this pump that's called the heart. You don't hear a lot about the lymphatic system. Well, the lymphatic system is tubing, essentially, that runs throughout your entire body. And very importantly, these tubes have valves. Now, fluid leaves the circulatory system, leaves the blood vessels, and it flows into the tissues to provide nutrients and oxygen to the tissues. The tissues are then surrounded by fluid in the extracellular space, and then that sort of has to get back into the circulatory system. Well, how it does this is it flows into the lymphatic system, this tubing that runs throughout our body. Now, these tubes have little valves, so when you that only lead in one direction. So fluid can go up the lymphatic tube, but it can't go back down the lymphatic tube. So what drives the lymphatic system? Well, actually, it's more your muscles and your movement. So as you walk or tense your muscles contract, which squeeze the lymphatic vessels, the lymphatic tubing, and that forces the fluid in one direction, because remember, those valves doesn't let it flow back in the other direction. And then when your muscles relax, it opens up the tubing, which allows more fluid to come in, and then as your muscles contract, it squeezes the tubing, forcing the liquid back up again. And this is why when you do a long haul flight, 8 hours or so, your ankles can swell up as the fluid, as you haven't contracted your muscles, so you haven't powered your lymphatic system. So the extracellular fluid just builds up in your ankles, which is super gross. What happens is, well, essentially the lymphatic fluid flows and flows and flows until it empties back into your blood vessels, into the large vein. It's called the vena cava, just above your heart. And so the lymphatic fluid can flow back into the circulatory system. But before it does that, it goes through lymph nodes. Now, lymph nodes are the little bumps under your arms and under your neck that doctors feel for when you're sick. And that's because when you're having a large immune response, your lymph nodes become very big. Now, inside your lymph nodes are packed full of immune cells, so it allows your immune system to evaluate what's going on in the extracellular fluid. Is there any pathogens going on there? But I'm going to jump into that a lot more when I talk about the adaptive immune system, because that's where the lymph nodes really come into it. But today in this video, we're talking about the innate immune system. Where else are immune cells? Well, they're in tissues. They just sit there in tissues, and we call these resident immune cells, in particular the epithelial tissues, the ones close contact to the non-internal parts of your body, so your intestinal lining and your skin and your mucosa in your mouth and that stuff, because they're going to be exposed to lots of pathogens. So there's loads of resident immune cells in there, also in your lungs, but also throughout your whole body. You could get an infection anywhere, so there are resident immune cells in your brain, for example, called microglia, but don't worry about that. They're awesome though. All right, so let's start with the circulatory system. Let's say we take some blood. Then we centrifuge it. Now, centrifuging is just spinning something really quickly, and it experiences centrifugal force, right, which pulls down cells and leaves the fluid component up the top there. So it pulls down heavy things to the bottom of the tube. So we end up with something like this. Now, early scientists noticed that your blood spins down and you end up with a fluid on top. We call that the plasma. That's the fluid component of your blood. And then the red blood cells all spin down to the bottom. But interestingly, less dense cells, so red blood cells are incredibly dense because they're packed full of protein and iron, and they don't have a large fluid cytoplasm. So they are the heaviest, so they spin down first. And then we end up with what we call a buffy coat, which is literally a white layer of cells. Now we call those cells the white blood cells. So we've got red blood cells which contain hemoglobin and iron, which makes them red, and then we have the white blood cells that do not contain that. Those are immune cells, those white blood cells. Now we call that the buffy layer or the buffy coat, and we noticed very early on in research that when you get an infection, this buffy layer increases, suggesting that those white blood cells had something to do with your immune response. Now, we call them leukocytes, and I think leuko is Greek or Latin for white, and cyte means cells. And you might recognize leuk from leukemia, which is a cancer of your white blood cells. So white blood cells, aka leukocytes, these are your circulating immune cells. And they look like this when we stain them under an H&E stain. So the red blood cells here, we can see them, they don't have a nucleus, and you can kind of see that they don't have a nucleus. Then we have these much larger white blood cells that all have a fantastic morphology. They're just beautiful, and we'll go into why they look like that in a second. Now, to explain that, I do have to dabble in explaining what a hematoxylin and eosin stain is. This is what an H&E stain is. This is the most common stain used in clinical pathology, in histology, in research, in blood smears. We just use this stain because it's a great way to visualize the cells. Remember, cells are mostly transparent, so it's good to put a dye in there or two dyes in there to allow you to identify the cells and some of the structures. So let's go through this. Eosin is an acidic stain, which means that it stains bases. So we know that acids and bases react. Baking soda and vinegar react because baking soda is a base, vinegar is an acid. So acids and bases react. So eosin is an acidic stain, and so it stains bases. And hematoxylin is a basic stain, so it stains acids. So you can see why these work really well together. They stain different structures because they target different components of the cell. And a couple of examples here is the protein collagen is full of basic amino acids, so it's a basic protein. And DNA and RNA, of course, are deoxyribose nucleic acid. So they're acids, so they'll stain with hematoxylin. Just to briefly touch on how that works, this is more of a heuristic. So hematoxylin is basic. What does basic mean? It means that it accepts H+ ions. That's the definition of a base. That's the definition of an alkaline. So it will accept an H+ ion, making it positive, right? So it was a positive hydrogen ion. It was neutral. It accepts the H+ ion. Now it's positive. DNA acids are the opposite. They donate H+ ions. So when it gives off its H+ ion to the solution, to the watery solution, it gives it off. It now becomes negatively charged. And now you can see you've got a positively charged hematoxylin, a negatively charged DNA acids. Opposites attract, certainly in chemistry. I don't think in terms of personality, but anyway, opposites attract. So we're going to get a purple stain on your DNA because the positively charged hematoxylin is going to react with the negatively charged DNA acid. So if we go back and look at the stain, we can see that the nuclei of all these cells are kind of funky. They're cool looking, but they're all dark purple because they're staining in that hematoxylin. And we can see that none of the red blood cells have that purple stain in there, and that's because they don't contain DNA. Red blood cells have no nucleus, right? So there isn't that dark purple stain inside the red blood cells. Now, you'll notice some of these cells look a bit different to the others. They seem to be full of these granules. This one over here is also full of those granules. You can see they're full of these weird granules, either pink granules, dark purple granules, or small little neutral colored pinky purple granules here. And we call these with these each have a name. One is eosinophil, and that's because it fills up with eosin. Yeah, that's a good way to remember it. So eosins attract eosin, the stain. So it's called an eosinophil. So the pink stain is all throughout that cytosol there, and so it's full of eosin. Then we have a neutrophil. Then we have a basophil, and now the basophil is full of hematoxylin, and remember hematoxylin is a base, and so it's a basophil because it fills up with the basic dye hematoxylin, which is why it's called a basophil. Now, remember, hematoxylin is a base, so it stains acids. Eosin is an acidic acid, so it stains bases. But neutrophils don't stain with either of them, and that's because they're neutral, right? So eosinophils, the basophil here is full of acidic compounds, the eosinophil here is full of basic compounds, and the neutrophil is neutral, right? So that's why it's called a neutrophil. So that's the naming of these cells, and these cells all collectively are called granulocytes because they're full of granules, and it's a super important subgrouping of the innate immune cells circulating around your blood. So let's jump into each one of those. We've got the neutrophil. It's most of your white blood cells. Most of your white blood cells are neutrophils. And it's really interesting. That doesn't happen in other animals, but it does happen in humans. And there is a hypothesis going around that humans living in their cities with open sewers and really high density were some of the grossest animals on the planet, and so we needed to shift our immune system balance to these neutrophils. But that's an interesting theory because we typically have more neutrophils than most other animals on the planet as a percentage. The neutrophil is the first leukocyte to arrive at an infection. So if you get an infection at your skin, your resident immune cells that are in the skin will send out signaling molecules, that many of which are called cytokines, and that will attract circulating immune cells. And the first ones to arrive are those neutrophils. They are a phagocyte, so they will eat up the pathogen. That's one of their major weapons. But their main weapon, this is the big one, is their granules contain enzymes that are toxic, or enzymes that produce toxic compounds. And an example is neutrophils contain an enzyme that produces bleach, and that's to sterilize the area. And it also causes damage to your tissue as well. Having bleach on the inside of your body is obviously not ideal. And so neutrophils are kind of that really quintessential innate immune cell that causes collateral damage whilst wiping out the pathogen there. Really non-specific, but super toxic and can wipe it out. The eosinophil, honestly, it's a lot like a neutrophil. So it is much lower percentage of your leukocytes, somewhere between 0 and 6%. This does depend on what infections you've got going on in the body, which changes those percentages. And the granules contain cytotoxic compounds, so much like a neutrophil, but in particular, they contain neurotoxins, and that gives you a hint at what kind of pathogens they attack, right? So a malaria protist, a bacteria, a fungus, a virus, a neurotoxin's not going to do anything to those organisms. So one of the main organisms they attack is the helminths, the worms that have a nervous system that can be attacked by an eosinophil. So here we have our worm here, absolutely getting swarmed by eosinophils, and they are spraying toxic compounds on there. And this includes proteases, as well as things similar to neutrophil that produce bleach, but also as well as those neurotoxic compounds to really wipe out those worms, which is pretty sweet. There's now a lot of new research out there that eosinophils also help with viruses, which is interesting and unique as well. But just remember that what they're mainly known for is swarming and killing these worms. But neutrophils can do it too as well. Neutrophils are also pretty effective at wiping out these helminths. As you can imagine, bleach is pretty good at killing all pathogens, but eosinophils seem to be especially good at it. Now, basophils, these guys are beautiful. They got that really dark hematoxylin stain all throughout their cytosol. They're 0.5 to 1% of your leukocytes swarming around your blood there. Their granules contain histamine. Now, histamine is acidic, which is why their granules stain really purple with the basic hematoxylin. So this is interesting, and this differentiates them from the other two granulocytes. So eosinophils and neutrophils, their granules contain antipathogen, toxic compounds to wipe out any pathogen around. Basophils contain histamine, which isn't an antipathogen agent. It's a signaling molecule, and it signals for inflammation. Right, so we're going to jump into what that is in a later video, but you know, that swelling that you get, the redness that you get, immune cells rushing into your tissue, your blood vessels dilating, which makes your tissue red, and your fluid leaking out of your blood vessels, which makes it swell up. Histamine is one of the major players in that, and we all sort of know this, right? This woman is having hay fever, an allergic reaction to pollen, and we can see that her face is red and puffy and swollen and full of mucus, and that's because of that histamine signaling molecule. And that's why you take antihistamines to get rid of hay fever. It blocks the histamine signaling, so it calms down that inflammatory process. So basophils, unlike the other two granulocytes, its granules contain signaling molecules to induce inflammation rather than toxic compounds to wipe out the pathogen. Now, we've got another funky cell in here. This is another innate immune cell, and it's called the monocyte, and it is massive, right? It's 2.8% of your leukocytes. They are phagocytes, so they're a big eater. They go in and they eat up the bacteria or fungus to try and destroy it. But they are also really important in coordinating the immune response. And to do that, they use these signaling molecules called cytokines, as well as other signaling molecules. But cytokines are a protein signaling molecule, which means you need to turn on some genes to create some mRNA to create the protein that is the cytokines, which will then be released and act as signaling molecules to all the other cells of the immune system and the vasculature to induce an immune response. So the monocytes are really like the conductor. They are orchestrating the entire immune response, particularly the innate immune response, with these signaling molecules, along with the basophil releasing that histamine, right? Now, I touched on a previous video what antigen presentation is, and I explained that all cells present antigens. All cells reach into their cytosol and display some of the biological molecules from their cytosol on the surface. So then an immune cell such as the T cell can come along and check to make sure that the protein in your cytosol isn't a pathogenic one. So if you're infected with a virus, and you're a cell, you would grab a viral protein, display it on your surface. A T cell could then come along and check it to see, is it self or is it pathogenic? Is it from outside the body, right? And induce that response. Well, there are some immune cells, particularly innate immune cells, that are professionals at this, right? And it's funny to think of professional, like they're not getting paid or anything like that, but they are a very specialized process to maximize this. So rather than just reaching into their cytosol to grab an antigen and display it, they're actually intentionally eating pathogens, right? They're a phagocyte. So they're going to intentionally eat pathogens to grab some of that and display it to the other immune cells like T cells. So they are a professional antigen presenting cell. Don't worry if this is all overwhelming. I'm going to be running through this all again, but as a process rather than breaking it down by cells. I just want to meet the players, meet the cells that we're going to talk about, and then in a future video I'm going to tie it all together. How do these cells all interact to coordinate an immune response? Now, the rest of the white blood cells are lymphocytes, and these are 20 to 40% of your leukocytes, and they have this, they're mostly nucleus. They have this giant nucleus and a very small cytosol, and they're primarily made up of T cells and B cells, which are cells of your adaptive immune system, and I'll cover those in the next video. So on to more innate immune cells. So we've done the circulatory innate immune cells. We've done the circulating immune cells that we call leukocytes. Those are our white blood cells. Now let's have a look at some of the other cells. So there are cells that sit in your tissue, waiting for pathogens to come into the tissue, and they're constantly surveying the extracellular environment. And one of the major ones here is a macrophage. Now it's this guy here. If we have a look, this is a lymphocyte. That's a macrophage, and you can see where the term macro comes in. So macro means big and phage means eater, and that's a macrophage there. So they are a phagocyte. They eat, they're constantly eating stuff in the extracellular environment. and they are massive. Remember, phage means eater, from phagocytosis. That's what they're doing. They're constantly phagocytosing. And we know it's to do with eating because esophagus, this P-H-A-G, means eating. So what I want to do to get an idea of what the macrophage does, let's compare the macrophage and the monocyte. So the monocyte is a circulating leukocyte. They are phagocytes, but they also coordinate the innate immune response and some of the adaptive immune response with cytokines. And they're a professional antigen presenting cell. They have a whole machinery designed around doing that professionally, right? Presenting the antigens from what they eat, the pathogens that they eat. The macrophage is a resident innate immune cell rather than a circulating one. They're a phagocyte and they coordinate the immune response with cytokines, and they're also a professional antigen presenting cell. So you can see that the macrophage really is the tissue resident form of a monocyte. And actually, kind of uniquely, a monocyte, once it leaves the blood vessel into the tissue, it can get attracted into the tissue to coordinate the immune response there. It can actually turn into a macrophage. So monocytes can turn in and replenish and replace the macrophage in a tissue, right? So you can think of these cells as basically the same cell. One of them is a circulating one, the monocyte. The other one is a tissue resident one, the macrophage. There's another cell that sits in our tissue, and that's a mast cell, and it particularly sits in the epithelial tissues. It is a granulocyte, and its granules are full of histamine. Now that might sound familiar. Basophil, mast cell, circulating granulocyte, the granules contain histamine. Histamine is acidic, which is why it soaks up that basic hematoxylin stain. And mast cells are the same. They're a resident innate immune cell granulocyte. Their granules contain histamine, and their histamine is acidic. Now, unlike the monocyte macrophage relationship, these guys have separate lineages. So a basophil cannot turn into a mast cell, but they are basically two sides of the same coin. The basophil is a circulating histamine containing cell, and the mast cell is a resident histamine containing cell. Right, up next I'm going to cover the cells of the adaptive immune system, and then in future videos I'll release next week, we'll look at how this all ties together. How can we tie the pieces of the puzzle together to see how your immune system responds in a coordinated fashion to wipe out the pathogens?